What's the first thing to happen when the samples show up at the lab? Well, they will generally arrive in large bags of samples that are collected from different sites. The laboratory takes those samples and scans them in, accessions the samples into the system, and then racks them. And they put them onto, each tube goes into a well on a 48 well plate, and that then gets transferred over to our robotic system. So our robotic system will take the 48 caps off of the tubes all at one time in about 10 seconds. That will get transferred over to another robot, which will pipette the saliva out of the tubes and onto a plate. The plate is the way we actually process the samples the rest of the way through the laboratory. Once that plate is filled with 96 samples, we seal the plate, and then we can take it out of our safety cabinet environment. These are potentially infectious samples, so they have to be handled in what we call a BSL 2+ environment. We assume that every sample is potentially infectious. But once it's sealed and it's taken out of that safety cabinet, we put it onto what we call a thermocycler. A thermocycler is basically a fancy way of heating up the samples. And we heat those samples to 90 degrees Celsius for 10 minutes. They're just short of boiling for about 10 minutes, and that does two things for us. That first completely inactivates the virus. We've tested this with local laboratories, and it's very effective and completely inactivates it, makes the sample safe to work with outside of the safety cabinet environment. The second thing that it does is it exposes the genomic material of the virus for the assay, and which is very critical. It really would not work as well if we weren't able to do this step as efficiently as we do. And basically make the material of the virus available for the PCR test to follow. Once that heating step is done, it goes to another laboratory where we transfer the samples over to another plate to begin the assay process. And the first step of that assay process is to amplify up all of the material in order to be able to put it onto the Fluidigm platform. Once that step is done, we take it to the final laboratory where we transfer the samples over to what we call the IFC, or Integrated Fluidic Circuit, which is a very cool thing that Fluidigm has built that really no one else has done. These IFCs allow us to put up to 192 samples on a single controller and up to 24 assays that we can run them against. So we can actually do 4,000 assays on this one plate. Which is part of the throughput provides part of the throughput of the capabilities of the system. Once that occurs, it goes on to the Fluidigm, the Biomark platform itself, to do the actual PCR, and then we get a digital readout, and we then take that into our LIMS system where we call the sample as positive or negative. The entire process takes approximately 3.5 to 4 hours from start to finish.